Good evening. My name is Cam Burton, and I was a researcher in the 2020 BSURPS program cohort. I worked in the Mudgett lab and my project involved looking at transcriptional responses in NHP-induced SAR. Now, that was a lot of jargon, so before I get started and get into the meat and potatoes of the project, I need to give you some background. So what is SAR? SAR, or Systemic Acquired Resistance, is a whole plant defense response triggered in response to a pathogen attack. During SAR, mobile signals are sent from infected tissues to uninfected tissues to turn on defense responses to prepare healthy tissues for possible new infections. This is known as defense priming. Recently, N-hydroxypiplicoic acid, otherwise known as NHP, was reported to be the key mobile signal that is required and sufficient to initiate SAR signaling. So here we see a visual representation of that. We see that our red pathogen here infects a local leaf tissue, which causes this mobile signals to be sent to this distal leaf tissue. We see then that this distal leaf tissue is now primed for defense against this pathogen, even though the infection happened in this local leaf. I was interested in seeing which genes were up and down regulated during this process to better understand what the plant is doing to initiate and to allow SAR to happen. To do this, I looked at transcription factors. What is a transcription factor? A transcription factor is a protein which regulates gene expression. So we can have transcription factors which promote gene expression and transcription factors which inhibit gene expression. Specifically, I looked at the Werke, NAC, BZIP, and BHLH families in Arabidopsis thaliana plant, which was the model system that I used to conduct this experiment. The wet lab portion of this experiment was done by my graduate mentor, Jessica Ferret. Essentially, she took Arabidopsis thaliana seedlings and treated one group with a mock treatment and the other with an NHP treatment. After this, RNA sequencing was used to measure the levels of RNA in the seedlings at the 15, 30, 180, and 360 minute time points. After this, we did a computational analysis to determine which transcription factor families were up and down regulated. This is where I come in. This is a flowchart of the methods that I used to do my computational analysis. First, I loaded the RNA sequencing data into R. Second, I conducted a differential gene expression analysis using the likelihood ratio test. A differential gene expression analysis is simply a way of quantifying how much a gene is upregulated or how much a gene is downregulated compared to a mock treatment. After this, I filtered significantly differentially expressed gene families of interest to prepare for clustering. These families were the aforementioned Werke, NAC, BZIP, and BHLH families. Next, I created gene expression clusters and heat maps to better understand these expression patterns. And finally, I ran the gene clusters through a gene ontology or GoTerm analysis using the TARE website. So what were my results? As we can see, I created these gene expression clusters for the Werke, NAC, BZIP, and BHLH families. From this, I chose to focus on these three Werke clusters and the first clusters of the BHLH family. The reason for this is that they had the most obvious level of difference between the NHP and the mock groups. As we can see, the cluster 1 of the Werkes were significantly downregulated in our NHP genes, while they were upregulated in cluster 2 and cluster 3 of our Werke genes. And then we can also see that in our BHLH cluster 1, we have a significant downregulation of this gene family in our NHP treated group. To focus in on these, I created heat maps where we can view individual genes within these clusters and see what their expression is relative to the mock. Here we see, for example, Werke 36, it was in cluster 1, and in our mock treatment, we see that Werke 36 is relatively upregulated, while in our NHP treatment, it is severely downregulated, with our red color being very upregulated and our blue color being very downregulated. So what does this all mean? Well, from my analysis and from putting these genes through my gene ontology analysis, I found out that the worky genes involved in our cluster 1 genes, which remember were significantly downregulated, were involved in flowering, petal expansion, and petal differentiation. Workies in cluster 2, which were upregulated, were involved in defense response to pathogens. Workies involved in cluster 3, which as you remember were upregulated, were also involved in pathogen defense as well as defense against abiotic factors. Our BHLH cluster 1 genes which we remember were significantly downregulated, were involved in cell maturation and differentiation. Based on this, I've concluded that during SAR, genes that are related to flowering, growth, and expansion and differentiation are in general downregulated, while genes related to defense are upregulated. This is consistent with what I knew about SAR going into the project. Based on this conclusion, we can say that a simpler way to put what I just said is that when a plant is involved in systemic acquired resistance in defense against a pathogen, it makes a trade-off between growth and defense. When defense is needed, growth is reduced, and when growth is needed, defense is reduced. So from this, some further areas of studies that I think that would be useful in better understanding SAR would be to look at other gene families besides the ones that I considered in my analysis today, and had I had more time, I would have definitely done so. So that concludes my project and my conclusions. I hope that it was very interesting to you. Before I end my presentation, I would like to give a big thanks to the BSURPS program as well as the VPU program for funding my research for the summer. I'd also like to thank the Mudgett Lab, specifically my graduate mentor Jessica Fourette and my other mentor Junggun Kim, as well as Mary Beth Mudgett, who is both my advisor and was my faculty mentor for the program. Thank you very much for listening, and have a wonderful day.